Namaste. Welcome to another session of the Deep Dive. Today, we're embarking on an incredibly important and really fascinating journey into a core biological process. How do organisms reproduce? This isn't just a vital chapter for our Class Chem board exams, It's also a fundamental concept. Forms the basis for lots of questions in competitive exams like NEET, JG, UPSC General Science, SSC, and even state PSCs. I'm definitely ready to unravel all its complexities with you.
Namaste to you too, and to all our dedicated learners tuning in. You've perfectly set the stage. Understanding reproduction, well, it goes far beyond just memorizing facts for exams. It's really about grasping one of the most fundamental processes, the one that ensures the continuity of life itself. In this deep dive, we'll systematically explore both the why and the how of reproduction. We'll move from the basic cellular mechanisms right up to the intricate human reproductive systems. Our goal here is absolute clarity, making sure you not only understand these concepts thoroughly, but can also confidently tackle any question that comes your way.
Okay, sounds great.
So let's begin with maybe a thought-provoking question, one that perhaps many of us just take for granted. Why do organisms reproduce at all? Think about it carefully. Reproduction, unlike, say, essential life processes like nutrition, respiration, or excretion, well, it isn't vital for an individual organism's survival. A single plant can complete its entire life cycle without producing seeds, right? And an animal can live its full lifespan without having offspring. In fact, creating new individuals actually demands a significant amount of energy that the parent organism could otherwise use for its own growth or repair or even self-defense. So why would an individual expend such precious resources on something that isn't directly vital for its own existence?
That's a really great point. If individual survival isn't the main driver, then it sounds like the main reason for reproduction is just to prevent the species from simply disappearing. Is it kind of a fundamental trade-off where individual energy is invested for the continuation of the entire species?
Absolutely. You've hit upon the core reason. Exactly. While reproduction isn't for individual survival, it is absolutely critical for the continuation of the species. If a species didn't reproduce, well, its members would eventually die out, wouldn't they? And that species would simply cease to exist on Earth. Consider this. We notice organisms. We perceive them as a distinct species because they exist in large numbers. If there were only one, maybe non-reproducing member of a particular kind, it's highly doubtful we would even be aware of its existence in the vast ecosystem. It's the sheer population size of a species that makes it noticeable, you see, and allows it to effectively fill its ecological niche. And how do we define a species in this context for our exams? Simply put, it's a group of organisms that look remarkably similar to each other and, crucially, are capable of reproducing amongst themselves to produce fertile offspring. So reproducing organisms essentially create new individuals that look very much like themselves. They ensure their genetic legacy and continued presence in the grand tapestry of life.
Okay, that makes sense. Species continuation is the key.
Precisely. Now that we've firmly established why reproduction happens, let's delve into the mechanics of how it happens. A fundamental question here is, do organisms create exact, perfect copies of themselves? You've observed that organisms of the same species look remarkably similar. That's obvious. This similarity in appearance is because their underlying body designs are fundamentally alike. And if the body designs are to be similar, well, then the fundamental blueprints or instructions for creating these designs must also be very similar, right? This brings us to the most basic event in reproduction, making copies of these biological blueprints.
Blueprints. You mean DNA, right? From Class 9.
Exactly. You got it. From your Class 9 biology, you'll recall that chromosomes, those thread-like structures in the nucleus, carry the crucial information for the inheritance of features from parents to the next generation. This information is meticulously stored in the form of DNA, deoxyribonucleic acid molecules. The DNA within the cell nucleus is essentially the master information source. It holds all the intricate instructions needed for making proteins. These proteins, in turn, are the workhorses. They're responsible for constructing and dictating the entire body design and all the functions of an organism. Therefore, logically, any significant change in this DNA information would lead to different proteins being made, which would ultimately result in altered body designs or functions. Makes sense?
Yes, clear. DNA holds the instructions for proteins, which determine body design.
Perfect. So, the most basic, the most fundamental event in reproduction is precisely the creation of a DNA copy. Cells achieve this remarkable feat by using complex biochemical reactions to build remarkably accurate replicas of their existing DNA. This intricate process results in two copies of the DNA residing within a single reproducing cell. Now, simply pushing one copy out of the cell wouldn't work, would it? Because that new DNA copy would be just a raw blueprint, completely lacking any organized cellular structure or organelles or the machinery needed to maintain life. Imagine having a detailed architectural blueprint for a magnificent house, but no bricks, no cement, no tools, and no builders to construct it. It wouldn't work.
Right, you need the whole setup, not just the instructions.
Exactly. Therefore, DNA copying is always accompanied by the creation of additional cellular apparatus. This means the cell also duplicates its vital organelles like mitochondria, its cytoplasm, all other necessary components, and then, only then, do the two DNA copies separate, each new copy taking with it a complete set of its own cellular apparatus. Effectively, this entire elaborate process culminates in a single parent cell dividing to give rise to two complete, fully functional daughter cells.
Okay, so DNA copies and the cell machinery copies, and then the cell divides into two.
Precisely. That's the sequence. Now, here's where it gets really fascinating and crucial for understanding evolution. Are these two resulting daughter cells absolutely identical to the parent, or even to each other? The simple answer is no. While they are remarkably similar, they are rarely, if ever, absolutely identical. This is because no biochemical reaction, no matter how precise, is perfectly reliable. There's always an inherent, albeit very small, chance of errors or variations occurring during the copying process. Think of it like making 1,000 photocopies of a document. Even with the best machine, you might find a tiny speck or a slight imperfection on one or two copies, you know?
Small errors creep in.
Exactly. So it's entirely expected that the process of copying DNA will introduce some minor variations, or what we might call errors, each time it occurs. As a result, the DNA copies generated will be similar to the original, but they may not be perfectly identical. The outcomes of these variations can differ significantly. Some variations might be so drastic, so fundamentally flawed that the new DNA copy simply cannot function correctly with the cellular apparatus it inherits. In such cases, that newly formed cell will unfortunately just die. However, there are many other variations that are subtle. These subtle variations don't lead to such a drastic outcome. The cells survive, and they are similar to the parent cell, but subtly different. This inherent tendency for variation during reproduction is the very basis for evolution, a concept that underpins all of biology, and one we'll definitely explore further in our upcoming deep dives.
I understand DNA copying is essential, and variations are a natural part of that, but why is this variation important? Isn't it better to make exact copies? Why would nature favor variation?
That's a truly critical question, and it's frequently asked in competitive exams because it connects this seemingly simple act of reproduction to the broader existential concept of species survival. Let's delve directly into the profound importance of variation. Organism populations thrive by filling well-defined places or niches within an ecosystem using their reproductive capabilities. The consistency of DNA copying is indeed important, yes, for maintaining the specific body design features that allow an organism to exploit that particular niche effectively. Reproduction, therefore, is intimately linked to the stability of populations of a species within its specific environment.
Okay, so consistency helps organisms fit their niche.
Correct. However, here's the crucial point. These niches are not static. They can change, and often for reasons completely beyond the control of the organisms themselves. Think about it. Temperatures on Earth can fluctuate dramatically over time. Water levels can rise or fall due to climate shifts. Or, to consider an extreme example from geological history, there could be a sudden meteorite impact that drastically alters the global climate. If a population of reproducing organism is perfectly suited to a particular niche, and that niche is suddenly and drastically altered, well, the entire population could face extinction.
I see. If everyone is identical and the environment changes, everyone suffers.
Exactly. This is precisely where variation becomes a lifesaver for the species. If some variations, even subtle ones, are present in a few individuals within these populations, there's a chance for those variants to survive the altered conditions. Let's take a classic example, often cited. Imagine a population of bacteria living happily in temperate waters. Now, if the water temperature were to suddenly increase significantly, perhaps due to global warming, most of these bacteria, adapted to temperate conditions, would unfortunately die. However, if by chance a few individuals within that population possessed a variation that made them resistant to heat, maybe a slight modification in their enzymes or cell membranes, these heat-resistant variants would survive. They would then reproduce, and their numbers would grow, ensuring the continuation of the species, albeit with a new, heat-resistant trait becoming prevalent.
So variation provides options for survival when things change.
Precisely. So, to summarize, Variation is profoundly useful for the long-term survival of the species as a whole. It ensures its adaptability and resilience to changing environments. It's nature's built-in mechanism for species to cope with unforeseen challenges and evolve. It's important to clarify, though, that variation is beneficial to the species, but not necessarily for every individual. As we discussed, some drastic variations can actually lead to the death of an individual organism. This critical distinction between individual and species benefit is a key concept frequently tested in your competitive exams, so internalize it well.
Right. Good for the species long term,
Yeah.
even if some individuals don't make it.
Yeah. Excellent. Now that we understand the fundamental why and the crucial role of DNA in variation, let's transition into the diverse how, the specific modes of reproduction. We'll start with modes of asexual reproduction. These are employed by single organisms or parts of a single organism. In asexual reproduction, new generations are created from a single parent individual without involving another parent. The specific mode an organism uses largely depends on its unique body design and level of complexity.
Okay, asexual reproduction. Single parent was first.
Our first mode is fission. For unicellular organisms, simple cell division itself directly leads to the creation of new individuals. It's very straightforward, very efficient. We observe many different patterns of fission. Many bacteria and protozoa, for instance, simply split into two equal halves during cell division. Let's first talk about binary fission. A classic example, often studied, is amoeba. In amoeba, the splitting of the two cells during division can essentially take place in any plane. Imagine one roti dividing perfectly into two equal rotis, a simple, clean split. That's binary fission in amoeba. However, some unicellular organisms show a bit more internal organization. Consider Leishmania, the protozoan that causes Kala-Azar. Leishmania has a distinct whip-like structure, a flagellum at one end. In such organisms, binary fission occurs in a very definite and precise orientation, specifically in relation to this flagellum. This ensures that each daughter cell receives the necessary structures in an organized way.
Okay, so amoeba splits anyway. Leishmania splits in a specific direction because of the flagellum.
Correct. Then we have multiple fission. This is seen in other single-celled organisms like Plasmodium, the infamous malarial parasite. In multiple fission, a single parent cell divides into many daughter cells simultaneously inside a protective cyst. So instead of one cell splitting into two, imagine one large gulab jamun producing many smaller ones inside its core before finally bursting open, releasing all the new, smaller gulab jamuns. That's analogous to multiple fission, creating numerous offspring at once, often under unfavorable conditions where the parent cell offers protection.
Tutor speaker. What's the core difference between binary fission and multiple fission? How can I remember it easily for the exam?
Excellent question for quick recall. The core difference lies in the number of daughter cells produced and the timing. In binary fission, a single parent cell divides to produce exactly two daughter cells. Think bi as in bicycle, meaning two. This division is often a relatively quick, direct splitting. In multiple fission, a single parent cell divides repeatedly within itself to produce many daughter cells simultaneously before releasing them. Think multiple for many. This often happens under challenging conditions where the parent cell forms a protective cyst. So two versus many is your quick differentiator.
Two versus many. Got it. Binary is two, multiple is many.
Perfect. Moving on to our next mode, we have budding. We observe this in activity 7.1 with yeast, remember? Small outgrowths, or buds, form separate from the parent and then grow further. A more intricate example is seen in Hydra, a simple aquatic animal. In Hydra, spiralized regenerative cells are used for reproduction through budding. A bud develops as a distinct outgrowth on the parent body due to repeated cell division at a specific localized site. These buds then progressively develop into tiny individuals, complete with mouth and tentacles. Once they are fully mature and capable of independent existence, they detach from the parent body and grow into new, independent organisms. You can think of it like a small plantlet growing directly from a parent plant and then becoming independent once it's strong enough.
Like a little mini Hydra growing off the side and then popping off.
Exactly like that. Our third asexual mode is fragmentation. This is exclusively observed in multicellular organisms that have a relatively simple body organization. A great example, frequently discussed in your syllabus, is Spirogyra. It's a filamentous alga, basically a long, unbranched chain of cells. When Spirogyra reaches maturity and its body is fully developed, its long filament simply breaks up into smaller pieces or fragments. Each of these fragments then has the remarkable ability to grow and develop into a completely new individual organism. This simple method works because each part of Spirogyra's body is largely self-sufficient. The cells aren't highly specialized into complex organs.
Why can't complex multicellular organisms like us reproduce by simple fragmentation or just by cell division? That seems efficient.
That's a very insightful question, and it's a common point of distinction in competitive exams, so pay close attention. The reason complex multicellular organisms like humans or even large trees and complex insects cannot reproduce by simple cell-by-cell division or fragmentation is fundamental to their body design and organization. Complex organisms aren't just random collections of cells. As we've learned, they exhibit an exceptionally high degree of organization. Specialized cells form specific tissues. Tissues organize into distinct organs, each doing a specialized job. Furthermore, these organs are precisely placed at definite positions within the body, forming an intricately coordinated system.
Right. Everything has its place and function.
Exactly. In such a meticulously organized situation, where different cell types perform highly specialized functions, nerve cells transmit signals, muscle cells contract, blood cells transport oxygen simple, random division or fragmentation would be utterly impractical. Chaotic, really. It simply wouldn't result in a viable, functional organism. Imagine if a small piece of your skin could grow into a whole new person. It can't. Skin cells are highly specialized. They don't contain the complete genetic blueprint or the necessary developmental machinery to form an entire, complex human being with all its intricate organ systems.
So specialization prevents fragmentation in complex organisms.
Precisely. Therefore, a basic strategy that has evolved in multicellular organisms is that different cell types perform different specialized functions. Following this pattern, reproduction in complex organisms is also the function of very specific cell types. These specialized reproductive cells are unique because they are capable of growing, proliferating, and most importantly, making all other cell types and tissues under the right conditions. This is why complex organisms need more elaborate and specialized reproductive mechanisms.
Okay, that makes sense. What's next after fragmentation?
Our next asexual mode is regeneration. Many fully differentiated organisms, particularly simple animals like Hydra and Planaria, the flatworm, possess an amazing ability. They can give rise to new individual organisms from their body parts. This means if the individual is somehow cut or broken up into many pieces, a remarkable number of these pieces can grow into separate, complete individuals. For instance, if you cut a planaria into several pieces, each piece can grow into a whole new planaria. This phenomenon is known as regeneration.
Wow, like starfish regenerating arms.
Similar concept, yes, but Planaria takes it a step further. The mechanism behind regeneration involves specialized cells, often called regenerative cells. These cells can proliferate, multiply rapidly to form a large mass of cells. From this initial mass, these cells then undergo changes and differentiation they specialize to become various cell types and tissues, reforming the entire organism in an organized sequence. This is called development. However, it's very important to note this distinction for your exams. Regeneration, in the context of creating a whole new organism from a fragment, is not the same as reproduction in the usual sense for most organisms. Most organisms don't normally rely on being cut up to multiply. It's more accurately described as a developmental potential or a robust repair mechanism rather than a primary mode of multiplication.
So if I accidentally cut a lizard's tail, it grows back. Is that regeneration too? And why can't we grow a whole new human from a limb then? This seems confusing.
That's a fantastic point for clarification, often tested. Yes, the regrowth of a lizard's tail is indeed a form of regeneration. However, it's absolutely crucial to distinguish between regenerating a lost body part, like a lizard's tail, and regenerating an entire new organism from a fragment, like Planaria. A lizard regrows its tail, but its tail cannot grow into a new lizard. See the difference? This is fundamentally different from organisms like Planaria, where each piece can grow into a complete new organism.
Oh, okay. Repairing a part versus creating a whole.
Exactly. The reason complex organisms like humans cannot grow a whole new individual from a limb, despite being able to heal wounds and repair tissues, ties back directly to what we discussed about body organization. Complex organisms have an extraordinarily high degree of cell and tissue specialization. Our cells are highly differentiated. They've become specialized for a particular function, and in doing so, largely lose their totipotency. That's the ability to differentiate into all cell types that embryonic stem cells or the regenerative cells of simpler organisms possess. A cell from your limb, for instance, is programmed to be a muscle cell or a skin cell. It can't spontaneously remember or execute the entire complex blueprint for a whole human. This concept of differentiation and loss of totipotency is key. It distinguishes repair in complex organisms from reproduction via regeneration in simpler ones. It's a common competitive exam concept.
Got it. Specialization limits full regeneration in complex beings.
Precisely. Let's move on to vegetative propagation. This is a significant mode of asexual reproduction primarily used by plants. In this mode, parts of the plant like roots, stems, and leaves can develop into entirely new plants under appropriate conditions. This is a remarkable property in plants, unlike most animals where such a mode for whole organism reproduction is much less common.
So plants can grow new plants from bits of themselves.
Yes, exactly. And this property is extensively utilized in agriculture and horticulture. It's crucial for commercial farming. Methods like layering or grafting are commonly used to grow many economically important plants: sugarcane, roses, grapes, even ornamental plants. There are several key advantages to using vegetative propagation that are important for your exams. Firstly, plants raised this way can bear flowers and fruits much earlier than those produced from seeds. Quicker yields, you see. Secondly, it makes it possible to efficiently propagate plants that have naturally lost the capacity to produce viable seeds. Classic examples include banana, orange, rose, jasmine, huge benefit commercially. And thirdly, a major advantage. All plants produced are genetically identical or very similar to the parent plant. This means they retain all the desired characteristics: yield, quality, disease resistance, ensures consistency.
Faster growth, seedless plants, and clones of the parent. Those are the big benefits.
You've nailed it. We can observe this in various examples. Consider Bryophyllum. Buds in the notches along the leaf margin can fall onto moist soil and grow into new plants. Or take a potato. If you cut it, only pieces containing an eye or a bud, which are actually modified stems, will grow new plants. Even a common money plant. Cut stem pieces with at least one leaf and node, put it in water, it grows. A piece without a node usually won't.
Right, I've seen that with money plants.
For more to know, let's briefly touch upon tissue culture. This is an advanced technique, a sophisticated form of vegetative propagation. New plants are grown by meticulously removing a small piece of tissue, or even single cells, from the growing tip of a plant, like the meristem. Under sterile conditions, of course. Let's break this down step by step. First, the isolated cells or tissue are placed in a carefully prepared artificial nutrient medium. Provides everything they need. Here, they divide rapidly to form a small, undifferentiated mass of cells called a callus.
A callus, okay, like a blob of cells.
Exactly. Second, this callus is then transferred to another specialized medium. This one contains specific plant hormones, auxins, and cytokinins, crucial for promoting growth and differentiation into roots and shoots.
So hormones tell the callus what to become.
Precisely. And finally, once these tiny, complete plantlets have developed basic structures, roots, shoots, leaves, they're carefully placed in soil or another growing medium. This allows them to mature into full plants. The advantages: huge. Many thousands of plants from just one parent, relatively quickly, and crucially, grown in disease-free conditions. Vital for commercial agriculture, especially for ornamentals.
So if a question asks about the benefits of vegetative propagation, these points about faster growth, seedless plants, and genetic similarity are key, right?
Confirmed. Those are indeed the critical advantages most often asked about in exams. Faster growth, seedless propagation, and genetic uniformity. Make sure you can explain each.
Perfect. What's the last asexual mode?
Lastly, for asexual modes, we have spore formation. This is observed in many simple multicellular organisms. A prime example is the common bread mold, Rhizopus. If you look closely at bread mold, you see fuzzy, thread-like structures spreading. Those are hyphae. They're vegetative, mostly for absorbing nutrients, not direct reproduction. The reproductive parts are the tiny blob-on-a-stick structure standing upright. Those blobs are called sporangia. Each sporangium contains numerous specialized cells called spores. These spores are remarkably resilient. They're covered by thick, protective walls, shielding them from harsh conditions like dryness or heat. When the sporangia mature, they burst, releasing the spores. These spores are very light, easily carried by wind. If they land on a suitable, moist surface with food, they can germinate and develop into new Rhizopus individuals, starting a new mold colony.
How does reproduction through spores benefit the organism? What are its advantages?
Good question. The benefits are quite significant for survival and spread. Firstly, spores are incredibly light and easily carried by wind, water, etc. This allows for wide dispersal, helping the organism colonize new places far from the parent. Secondly, the thick, protective walls are a major advantage for survival in adverse conditions. They allow spores to stay dormant, withstand harsh environments, until conditions become favorable, especially moisture. And thirdly, organisms using spores can produce them in vast numbers, huge quantities. This dramatically increases the chances that at least some spores will land somewhere suitable and grow, maximizes reproductive success. So broad dispersal, protection, and high quantity are the key advantages.
Dispersal, protection, quantity. Makes sense for something like mold.
Exactly. Now, we shift our focus significantly from asexual reproduction involving a single individual to sexual reproduction. This mode fundamentally depends on the involvement of two individuals to create a new generation. We're all familiar with examples. Bulls alone can't produce calves. Hens alone can't produce chicks. Both sexes, male and female, are needed. This naturally raises a crucial question. What's the significance of this sexual mode? Are there limitations to asexual modes that sexual reproduction addresses better?
Right. Why involve two parents if one can do the job? What's the advantage?
Let's consider why the sexual mode is so prevalent, especially in complex organisms. We already discussed that DNA copying, while precise, isn't absolutely accurate. The resulting variations are fundamental for diversity. While these variations might not protect every individual, they are incredibly useful for the species' long-term survival, especially in changing environments. However, these natural variations arise relatively slowly through asexual reproduction. If the environment changes drastically and quickly, a new disease, a sudden climate shift relying solely on slow, random variations might not be enough for a species to adapt in time. So how can the process of making variants be sped up? The answer lies in sexual reproduction.
So sexual reproduction generates variation faster.
Exactly. Each new variation is built upon accumulated variations from previous generations. Therefore, two different individuals in a population will likely possess quite different patterns of accumulated variations in their DNA. Crucially, since these variations are in living, functional individuals, we know they don't have drastically harmful effects. Now here's the genius part. Combining DNA from two different individuals, each carrying unique variations, creates entirely novel combinations. Each resulting combination in the offspring is completely new and unique. It involves DNA from two distinct parents. The sexual mode inherently incorporates this process of combining DNA from two different individuals. This significantly speeds up the generation of new variations. Accelerated diversity is vital for adaptability, driving evolution, allowing species to respond better to rapid changes.
Okay, combining DNA from two parents creates new combinations of traits much faster. Big advantage.
Huge advantage. But this combination presents a major biological difficulty. If each new generation simply combines the full DNA from two parents, then each new generation would have twice the DNA amount as the previous one.
Oh yeah, that would double every time. That can't work.
Exactly. It would quickly disrupt cellular control, lead to non-functional cells, non-viable species. So how has nature brilliantly solved this fundamental problem?
How did it solve it?
We observed earlier that as organisms get more complex, tissue specialization increases. One brilliant solution evolved by many multicellular organisms is having special lineages of cells, germ cells, in specialized reproductive organs. These germ cells undergo a unique process of cell division called meiosis.
Meiosis, I remember that term.
Good. Let's take this step by step to understand meiosis properly, as it's often asked about in detail. First, within these specialized germ cells, meiosis occurs. It's a special type of cell division, different from mitosis used for growth. Second, during meiosis, the number of chromosomes, and thus the amount of DNA, are precisely reduced to half compared to normal body cells. For humans, 46 chromosomes become 23. And finally, when these haploid germ cells, or gametes, like sperm and egg, from two individuals combine during fertilization, the original correct chromosome number and DNA content are reestablished in the new generation, forming the diploid zygote. This elegant mechanism ensures DNA content remains constant across generations, while still allowing the crucial combination of variations.
So meiosis has the chromosomes in sperm and egg, so when they combine, the baby gets the normal number. Clever.
Very clever indeed. Furthermore, if the zygote is to grow into a complex organism, it needs substantial energy stores and structures. In very simple organisms, the two gametes might look similar, but as body designs become more complex, the germ cells themselves become highly specialized. Typically, one germ cell becomes large and contains significant food stores. This is the female gamete or egg. The other is usually smaller and motile, capable of active movement. This is the male gamete or sperm. The need to create these two different types of gametes has naturally led to the evolution of differences in male and female reproductive organs, and often noticeable differences in the bodies of males and females.
Two to speakers. So the biggest advantage of sexual reproduction is the increased variation, which helps in evolution. And meiosis is what prevents the DNA from doubling up every generation, correct?
Precisely. You've perfectly captured the two most critical takeaways regarding sexual reproduction and meiosis for your exams. Accelerated variation is the evolutionary engine sexual reproduction provides, allowing faster adaptation. And meiosis is the elegant solution to maintaining chromosome number and DNA content, ensuring stable offspring from combined genetic material. Understanding this interplay is absolutely key.
Great, that clarifies it.
Excellent. Now let's dive into sexual reproduction in flowering plants, specifically angiosperms. Very important for ecosystems and exams. The reproductive parts are all contained within the flower itself. You might recall parts like sepals, outer green leaves protecting the bud, and petals, often colorful to attract pollinators. But the crucial reproductive parts are the stamens, male, and the pistil, female, which contain the germ cells.
Stamen is male, pistil is female.
Right. Correct. Let's look closer. The stamen is the male part. It typically has a stalk called the filament, and at the tip, the anther. The anther is where the yellowish pollen grains are produced. You've probably seen pollen powder. These pollen grains contain the male germ cells, or male gametes.
Okay, stamen makes pollen, pollen has male gametes.
Exactly. Now the pistil, or carpel, that's the female part, usually in the center. It has three distinct parts, often vertically arranged. First, the ovary, the swollen bottom part, very important. It contains the ovules, and each ovule contains the female germ cell, the egg cell, or female gamete. Second, extending up from the ovary is the style, a slender middle part. And third, at the very top is the stigma. It's the terminal part, often sticky or feathery, designed to effectively trap pollen grains.
Ovary at the bottom with ovulus eggs, style in the middle, sticky stigma at the top. Got it.
Perfect. Flowers can also be categorized based on these parts. A flower is unisexual if it has either stamens or the pistil, but not both. Examples: papaya, watermelon flowers. A flower is bisexual if it contains both stamens and the pistil in the same flower. Examples: hibiscus, China rose, mustard flowers.
Unisexual is one sex. Bisexual is both sexes in one flower.
Correct. Now, the first crucial step in their sexual reproduction is pollination. Definition: transfer of pollen grains from the stamen, specifically the anther, to the stigma of a flower. It can happen in two main ways: self-pollination, pollen transfer within the same flower or to another flower on the same plant, leads to genetic similarity; cross-pollination, pollen transfer from one flower to a flower on a different plant of the same species. This transfer is typically helped by external agents: wind, water, or animals like insects, bees, butterflies, and birds, attracted by the flower.
Self is same flower plant, cross is different plant, often needing wind or animals.
Exactly. After successful pollination, the next vital step is fertilization. Once a pollen grain lands on a suitable stigma, it germinates. A small pollen tube grows out. This tube travels down through the style, reaching the ovary. Inside the ovary, the male germ cell from the pollen moves through the tube and fuses with the female gamete, egg cell, inside the ovule. This fusion of male and female germ cells is fertilization. It results in the formation of a zygote, the first cell of the new plant.
Pollen tube grows, male gamete travels down, fuses with egg in the ovule. That's fertilization.
Precisely. Following fertilization, remarkable changes occur, leading to seed and fruit formation. The zygote divides repeatedly by mitosis, forming a multicellular embryo within the ovule. Concurrently, the ovule develops a tough, protective outer coat and becomes a seed. Meanwhile, the entire ovary surrounding the ovules grows rapidly and ripens, developing into a fruit. As these changes happen, other flower parts, petals, sepals, stamens, style, stigma, usually shrivel and fall off. Their job is done.
The zygote becomes embryo, ovule becomes seed, ovary becomes fruit, petals fall off.
Excellent summary. The formation of seeds offers significant advantages. The seed contains the future plant, embryo, protected by a tough coat. This provides dormancy, allowing survival in bad conditions, and facilitates dispersal. Seeds get carried far and wide. Finally, under appropriate conditions, moisture, warmth, oxygen, the seed germinates. The embryo develops into a seedling. You can see this easily by soaking Bengal gram seeds. They sprout, right? That's germination.
What's the difference between pollination and fertilization again? This is a common confusion point.
That's an excellent question to clarify, definitely a frequent source of confusion and tested often. Let's make it crystal clear. Pollination is simply the transfer of pollen grains from the anther to the stigma. It's a physical event, bringing the male gametes near the female part. Think delivery. Fertilization, on the other hand, is the actual fusion of the male germ cell with the female gamete, egg cell. This happens after successful pollination once the pollen tube delivers the male gamete. So pollination is the prerequisite transfer, fertilization is the subsequent fusion, distinct processes.
Transfer versus fusion. Okay, much clearer now.
Good. Now let's move to reproduction in human beings. Immensely important topic, biologically and societally, critical for your exams. Humans, like many complex animals, use a sexual mode of reproduction.
Right, involving male and female systems.
Exactly. We begin with something seemingly unrelated but fundamentally connected, how our bodies change as we grow. You learned about height increase, milk teeth replacement in class 8, general growth. But in our early teenage years, roughly 10 to 19, a whole new set of profound changes occurs. Not just body enlargement. Body appearance changes, proportions shift, new features emerge, new sensations, emotional shifts. This period of adolescence is called puberty. It's fundamentally when our reproductive tissues begin to mature, usually as general body growth slows down.
Puberty, the time of big changes in teenagers.
Correct. Some changes are common to both boys and girls during puberty. Thick hair grows in new places. Armpits, genital area, which can also darken. Thinner hair might appear on legs, arms, face. Skin often becomes oily, sometimes leading to pimples. And importantly, we become more conscious of our own bodies and others' bodies in new ways. New sensations, attractions, signal sexual maturation.
Hair growth, oily skin, more awareness.
Wait.
Common changes.
Yes. Then there are distinct changes specific to boys and girls, driven by different hormones. In girls, breast size typically begins to increase with darkening of the nipple skin. Crucially, girls also begin to menstruate around this time, marking the start of reproductive cycles. In boys, new thick facial hair, beard, mustache, often appears. Voices begin to crack and deepen. The penis occasionally enlarges and becomes erect, indicating maturation.
Girls, breast menstruation.
Boys.
Facial hair, voice change, penis changes. Got it.
Right. It's important to remember these changes are gradual over months or years. They don't happen all at once or at the exact same age for everyone. Some start early, progress quickly, others are slower. Each change also develops over time. Facial hair starts sparse, then gets uniform. There are individual differences too, just like nose shapes differ. Patterns of hair growth, breast, or penis size vary. All these changes are aspects of sexual maturation, signaling the development of reproductive capabilities.
So it's a slow, variable process signifying readiness for reproduction.
Exactly. But why does sexual maturation happen at this age? As we discussed, complex organisms have specialized cells. Creating germ cells, sperm, eggs, is highly specialized and energy-intensive. While growing to adult size, resources are directed towards general growth. Reproductive tissue maturation isn't the priority then. So, as general growth slows, resources become available and reproductive tissues mature. This period is puberty. These physical changes also act as outward signals of sexual maturity, important for mating in many species. Specialized organs like an erect penis or mature breasts are adaptations for germ cell transfer and nurturing offspring.
What specific changes are seen in girls at puberty, for example, for a 10th board exam question?
For your Class 10 board exams, the key changes to highlight for girls at puberty are, first, a noticeable increase in breast size, often accompanied by a darkening of the skin around the nipples. Second, and very importantly, the onset of menstruation. These 3 points are usually sufficient and accurate.
Breast size increase, nipple darkening, menstruation starts.
Okay. Let's now delve into the specific systems, starting with the male reproductive system. It's designed to both produce male germ cells, sperms, and deliver them efficiently.
Produce and deliver sperm. What are the parts?
The formation of sperms happens continuously in the testes. These are located outside the main abdominal cavity in a pouch called the scrotum. The reason for this external location, crucial. Sperm formation requires a slightly lower temperature, about 2-3 degrees Celsius cooler than normal internal body temperature. We also know testes secrete the hormone testosterone. Testosterone regulates sperm formation and brings about all those male puberty changes, facial hair, deep voice, muscle development.
Testes outside in scrotum for lower temperature needed for sperm. They also make testosterone.
Correct. From the testes, sperms travel through a long tube called the vas deferens. This vas deferens goes up and joins a tube from the urinary bladder, forming the urethra, a common passage for both sperm and urine. Along the path, 2 important glands, the prostate gland and the seminal vesicles, add their secretions. These secretions mix with sperms to form a fluid called semen. This fluid makes sperm transport easier, liquid medium, and provides vital nutrition to the sperms.
Vas deferens carry sperm, joins urethra. Prostate and seminal vesicles add fluids to make semen.
Precisely. The sperms themselves are tiny microscopic bodies designed for function. Each has genetic material, DNA, in the head, a midpiece with mitochondria for energy, and a long tail. The tail is crucial for motility, enabling the sperm to swim actively towards the female egg.
Could you explain the specific functions of the seminal vesicles and the prostate gland again? I sometimes mix them up.
Absolutely. Common exam point requiring precision. Both are accessory glands, vital for semen. The seminal vesicles secrete a fluid rich in fructose. Fructose is the primary energy source, nourishment for sperms, fueling their journey. Also adds volume. The prostate gland secretes a milky, slightly acidic fluid, also adds volume. Key functions: aids sperm motility, helps them swim well, and contains substances that protect sperms from the acidic female reproductive tract. Crucial for survival. So think nourishment from seminal vesicles, motility, and protection from the prostate.
Seminal vesicles, nourishment, fructose. Prostate, motility, aid, and protection. Got it.
Excellent. Now let's explore the female reproductive system, responsible for producing female germ cells, eggs, nurturing a developing embryo, carrying pregnancy.
Making eggs, carrying the baby. What are its parts?
Female germ cells, eggs, ova, are produced in the ovaries, two almond-shaped organs in the lower abdomen. Ovaries also produce female hormones, mainly estrogen and progesterone, regulating the menstrual cycle and female characteristics. Interestingly, a girl is born with thousands of immature eggs already in her ovaries, dormant. They only start maturing at puberty. From then on, typically one egg matures and is released by one ovary each month, alternating.
Ovaries make eggs, one per month after puberty, and female hormones.
Right. Once released, the egg is swept towards the womb, or uterus, through a thin tube called the oviduct, or fallopian tube. Two tubes, one for each ovary. The oviduct is the usual site where fertilization occurs, where sperm meets egg. The two oviducts unite into a larger elastic muscular bag, the uterus or womb. This is where the fertilized egg, embryo, implants and develops into a fetus. The uterus opens into the vagina through a narrow passage called the cervix. The vagina receives sperms during intercourse and is also the birth canal.
Egg released goes into fallopian tube, oviduct, where fertilization happens, then to uterus for development. Uterus opens via cervix into vagina.
Perfect pathway. Now, fertilization and embryo development. During intercourse, millions of sperms enter the vagina. They swim upwards through cervix, uterus, into oviducts. If an egg is present there, they may encounter it. Fusion of one sperm with the egg results in a fertilized egg, the zygote. The zygote immediately starts dividing rapidly, mitosis, forming a ball of cells, the embryo. This embryo travels down the oviduct and implants in the thick, blood-rich lining of the uterus. Implantation. Here, the embryo grows, cells differentiate, forming organs, becoming a fetus.
Zygote divides to embryo. Embryo implants in uterus, develops into fetus.
Correct. To support this, the mother's body is amazing. The uterus prepares monthly with a thick, spongy, blood-rich lining for the embryo. A crucial structure for nourishment is the placenta, a special disc-shaped tissue from both embryo and uterine wall embedded in the wall. Let's look closely. Structurally, it has finger-like villi on the embryo's side. These villi are surrounded by blood spaces on the mother's side, creates a huge surface area for efficient transfer. Functionally, it's the critical exchange point. Nutrients, glucose, amino acids, vitamins, oxygen from mother's blood to embryo. Conversely, waste products, CO2, urea, from embryo transfer to mother's blood via placenta for her systems to excrete. Development inside takes about 9 months, gestation. Birth occurs via rhythmic uterine muscle contractions.
How does the embryo get nourishment while it's developing inside the mother's body? That placenta seems key.
The placenta is absolutely the key. It's the embryo's life support system, the vital connection. It facilitates exchange without bloodstreams directly mixing. Firstly, nutrient transfer. Glucose, amino acids, vitamins, minerals from mother's blood. Secondly, oxygen supply. Oxygen diffuses across from mother's blood. Thirdly, excretory organ for the fetus. CO2, urea, diffuse from fetal to mother's blood for removal. And finally, the placenta acts as an endocrine gland, producing hormones like progesterone, crucial for maintaining pregnancy. So it handles respiration, nutrition, excretion, and hormone production for the fetus.
Wow, the placenta does a lot. Respiration, nutrition, excretion, hormones.
It really does. Now let's consider what happens when the egg is not fertilized. An unfertilized egg lives only about 1 day after release. The ovary releases 1 egg monthly. The uterus prepares each month, building that thick, spongy, blood-rich lining for a potential embryo, essential for nourishment if fertilization and implantation occurred. However, if fertilization doesn't happen within that roughly 1-day window, well, the prepared uterine lining isn't needed. The body then efficiently sheds this lining. It slowly breaks down. It's expelled from the body through the vagina as a mixture of blood and mucus. This entire cycle is menstruation. Typically occurs roughly every month, average 28-30 days, but varies. A period usually lasts about 2 to 8 days.
Why does menstruation occur? What is its biological purpose?
Re-emphasizing. Menstruation is the shedding of the uterine lining that was prepared to nourish a potential embryo. When pregnancy doesn't happen, the specialized lining is expelled from the body. It's the body's monthly reset of the uterus, preparing for a new cycle.
Okay, shedding the unused lining. Makes sense.
Finally, let's discuss reproductive health. A critical topic, extending beyond biology to societal, ethical, personal well-being. Relevant for UPSC too. As we've seen, sexual maturation is gradual, overlaps with growth. Reaching puberty doesn't necessarily mean the body or mind is fully ready for sexual acts or for having and raising children. This brings a significant point. How do individuals decide readiness? We all face pressures. From friends, maybe? From family or society to marry, start families. Conversely, maybe pressure from government campaigns for family planning. Making informed choices can be difficult.
Yeah, readiness isn't just about biology, it's mental and social too.
Exactly. We must also consider health consequences of sex. Diseases can transmit person to person. Sexual intercourse involves intimate contact. So many diseases can be sexually transmitted, STDs or STIs. Examples, bacterial infections like gonorrhea, syphilis, treatable with antibiotics if caught early. Viral infections like genital warts, HIV, AIDS, currently no definitive cure, compromised immune system.
Bacterial ones like gonorrhea, syphilis, viral like warts, HIV, AIDS. Serious stuff.
Very serious. Is prevention possible? Using a covering for the penis during sex, a condom, can significantly help prevent transmission of many infections. It's a physical barrier. Safe practices are crucial. Furthermore, sex always carries the potential for pregnancy. Pregnancy places huge demands, physical, emotional, financial, on the woman. If not ready, her health, the child's well-being, the family's well-being could be affected. Therefore, various methods exist to avoid pregnancy, contraceptive methods, or birth control. They fall into category.
Okay, contraception to avoid pregnancy. What are the types? One category, mechanical barriers. Physically prevent sperm reaching egg. Common examples, condoms for penis, similar coverings worn in vagina, diaphragms, cervical caps. Physical block, another category. Hormonal methods. Alter hormonal balance to prevent egg release, ovulation, or implantation. Common form. Oral pills taken daily by women. Highly effective, but can have side effects due to hormone changes, nausea, weight change, mood shifts. Then there are intrauterine devices, IUDs, small devices placed in the uterus by a professional. Examples. Loop, copper T work by preventing implantation or making uterus hostile to sperm, can sometimes cause side effects, irritation, bleeding. Lastly, surgical methods, more permanent, create blocks for gamete transfer, male. Vasectomy, vas deferens blocked, permanently stops sperm reaching urethra, female. Tubal ligation, fallopian tubes blocked. Stops egg reaching uterus, sperm reaching egg. Fertilization impossible, generally safe long-term, highly effective. But any surgery carries risks, infection, etc. if not done properly.
Barriers, condoms, hormones, pills, IUDs, copper T, surgery, vasectomy, tubal ligation. Lots of options. Yes. And it's important to mention a serious societal concern related to surgical methods for unwanted pregnancies, particularly in India. This includes the illegal, unethical practice of sex-selective abortion, specifically aborting female fetuses. This severely harms the natural female-male sex ratio. It's prohibited by law, PCPNDT Act, but sadly continues, causing alarming declines in child sex ratio. Crucial social issue for UPSC.
That's a very serious issue, the illegal sex-selective abortion.
Indeed. Finally, we noted reproduction increases populations. Population size depends on birth and death rates. Expanding human population is a concern for many harder-to-improve living standards, resource issues. It's complex, needs critical analysis for exams like UPSC. Is population the main issue, or is societal inequality a bigger factor in poor living standards? Worth thinking about.
Does using a copper T also protect against sexually transmitted diseases, or just prevent pregnancy?
That's absolutely correct to ask, and a critical distinction. A copper T, like other IUDs and hormonal methods, is designed only to prevent pregnancy. It works by stopping fertilization or implantation. It provides absolutely no protection against STDs. For STD protection, barrier methods like condoms are necessary as they physically block fluid exchange. Contraception and STD prevention are distinct goals, often needing different methods. Crucial to understand.
Okay, copper T is only for pregnancy prevention, not STD protection. Clear. Finally, tutor speaker, what are the main reasons why people adopt contraceptive methods?
Certainly, the main reasons revolve around responsible family planning and reproductive health. Firstly, most directly to prevent unwanted pregnancies, empowers individuals. Secondly, enables planning families deciding when and if to have children. Thirdly, facilitates spacing births, allowing maternal recovery, resource focus on existing children. Ultimately, contributes to the health and well-being of the mother and existing children, avoiding strain from unplanned pregnancies. So it's about empowerment, planning, spacing, and health. Crucial aspects.
Planning, spacing births, preventing unwanted pregnancy, overall health. Got it.
We've covered a vast and intricate landscape today in our deep dive into how do organisms reproduce. We started by understanding reproduction is for species survival, driven by DNA copying with inherent variation, the engine of evolution. We explored diverse asexual modes, fission, budding, fragmentation, regeneration, distinguishing it, vegetative propagation, including tissue culture, and spore formation for dispersal and survival. We transitioned to sexual reproduction, highlighting its role in speeding up variation by combining genetics. We understood how meiosis prevents DNA doubling. Our journey took us to sexual reproduction in flowering plants, flower anatomy, pollination, fertilization leading to seeds and fruits. Finally, we delved into human reproduction, puberty changes, male and female systems, fertilization, embryo development, the vital placenta, menstruation, and crucial reproductive health topics like STDs and contraception.
It's a lot, but seeing it all connected is helpful.
Exactly. Remember, understanding these processes isn't just for exams. It's about understanding life itself and making informed choices. To solidify this, revise all topics thoroughly. Practice past year questions, Class 10 boards, NEET, GE, UPSC, general science. Systematic practice reinforces knowledge and prepares you for any question type. And as always, please don't hesitate to reach out if you have any doubts on this chapter. Clarifying them is crucial for your success.
What an incredible deep dive, truly. It's fascinating to see how every detail, from DNA variations to hormonal shifts, connects to the grand story of life continuing. It really makes you appreciate the resilience and adaptability of living organisms, and how our own understanding helps us navigate this complex biological world. Keep exploring, keep questioning, and join us next time for another deep dive into the wonders of science.